Welcome to tonight's Labcast. I'm Nick Chatterton, I'm a member of the module team. So with me I have Eleanor Crabb, who's also going to help us go through tonight's session. Eleanor, what are we doing tonight?
In this particular Labcast, we're going to be looking very much at how we would analyse spectra, so looking at very much at the strategy you would use when kind of interpreting NMR spectra. We will be looking very much in terms of, say, focusing on the interpretation rather than the theory. So you should be aware of the fact that we have a slide cast on NMR, the fundamentals. So that's really where you would find all the theory that you would need. The important thing about that slide cast is you're not expected to remember the theory, but really the theory is there to support your understanding of kind of how we go about interpreting it and some of the key aspects. So in terms of the objectives, really, as I say, it's to kind of look at the strategies for interpreting NMR spectra. So we're going to be looking at things such as how we would determine the number of resonance lines. So that really relates to the environment of the nuclei in the particular molecule. We'll be looking at the chemical shifts, so where they occur in the spectrum. We'll also be looking, in the case of the proton NMR, about looking at kind of intensity of the signals and how we would use the integrals. But then also looking, again, very important, particularly in the proton NMR, is this idea of the coupling, so where we're looking at kind of coupling and how that affects the spectra in terms of multiplicity. So those really are kind of the main things that we'll be looking at during the presentation. We will also have a number of widgets, so these are where we can basically ask you questions to give you an opportunity to answer a few of the questions that we're asking as we go along. For those of you watching the recording, you obviously won't be able to interact with these widgets. However, you might like to look at the question and think about how you would actually go about answering these yourselves. And then obviously you're going to be able to see how everybody else has voted, and we will go over the answers anyway.
Okay, so basically what we're going to do tonight is run through some specific NMR spectra. Myself and Matt Kershaw, one of the NMR technicians, went into the lab and recorded the NMR spectra for you. So what we did when we were producing those spectra was a little video, which I'd like to just run through now to show you how we did this. So what we have there is the NMR spectrometer, newly refurbished here in Milton Keynes. So as I say, Matt was adding basically deuterated chloroform, so deuterated solvent, to a sample of ethyl benzene. So he's already got the ethyl benzene in there, and he's just adding in about 1 milliliter of deuterated chloroform. And obviously what he's going to do now is make sure that those two have mixed, and he's then adding, once he's mixed them, he then adds them into the precision glass of the NMR tube. So just about, as I say, between 0.7 and 1 milliliter of a solution. Okay, so if that's how we make up, very, very simply, how we make up the NMR sample, we then cap it and then take it across the spectrometer. So the first thing we do is need to make sure that it's at the right height, such that it sits in the magnet properly. So we're just doing that there. He then places it onto the carousel, and then it goes over to the computer and selects his sample. And the carousel goes around, and hopefully then picks up, uses some compressed air to pick up our sample, shoots up there, and then it goes to a nice robotic arm, passes it along, and then down into the core of the NMR machine. Okay, so that's kind of very sort of modern set of apparatus. Is then in the machine, in which we're running a proton NMR here, and that will take about maybe 1 to 1.5 minutes to run for a proton NMR. And then, as I say, to 400 megahertz there, as you can see written on it. So that's we'll use that later on. So it's 400 megahertz NMR machine. The sample's now finished, goes back into the carousel, and then we've got the picture of Matt there processing the spectrum for ethyl benzene. So he does various things such as integrate, so getting an idea of the intensity, looks at the multiplicity of the peaks, and also does stuff like get some expansions. So focuses on particular parts of the spectrum and blows those up so we can see their features more clearly, and we'll look at some spectra of that type later on in the Labcast. And we've got one expansion there. It's an expansion of the aromatic region of ethyl benzene, and we'll look at these, maybe not this one, but some other sort of splitting patterns during today's Labcast. Okay, so that's on the practical side. So what's next, Ella?
Okay, right. So what we're going to think about now is really to do with the chemical environment. So we're going to be considering the ethyl benzene molecule. And we thought we would start with carbon-13 NMR because obviously this should be more familiar to you because this is the one that you did at level 2. So we're looking at carbon-13 NMR. And we would like you to think in terms of the ethyl benzene molecule, how many different chemical environments you think we have for the carbon nuclei within that molecule. So we have
a picture.
A picture here of ethyl benzene. It's
hand-drawn, sophisticated-looking picture of ethyl benzene.
Indeed, and actually, if you look at the widget, we've also got a slightly more sophisticated version on the widget. So we would like you to indicate there. So thinking very much of the symmetry of the molecule and the different types of environments you would have for the carbon nuclei within that molecule, because I think you would have seen, hopefully from your studies at level 2 and also the fundamentals, the NMR fundamentals about the idea about environments relating to resonance lines. So we'll leave you to look at that widget, but in the meantime, Nick is going to look at the kind of the carbon environments for some di-substituted benzene.
Okay, so as Eleanor said, that's just to show you this is a so-called mono-substituted benzene because you've just got one group off the benzene ring. So let's just look at 2 di-substituted benzenes, partly because we'll need to look at these a bit later on in the labcast. And I'll start with a more symmetrical one. Let me just draw it out. So I'm going to have 2 chlorines, one at the top and one at the bottom. Okay, so that's fairly symmetrical. And by symmetrical, I mean you can kind of see that there's a sort of plane of symmetry there. Okay, so one half's the same as the other half, and there's also a plane of symmetry here, so that half is the same as that half. And that helps us identify how many different chemical environments there are. Okay, so if we think about it, we've got, let me just change colour pen, we can see that, for example, these two, this one is the same as that one, and it's also the same as that one, and that's to do with this plane of symmetry. This, and this are the same. So let's just mark those on. That one, the same as that one, the same as that one, the same as that one. So all 4 of those will give rise to one signal. But there's also, these two are obviously a little bit different because this is directly bonded to a chlorine and so is that one. So those 2 other carbon atoms are also in a different environment. So we have a second chemical environment, which we'll mark with orange. So with this one, we would expect to see a grand total of 2 signals in our carbon NMR spectrum. Okay, so that's a nice, highly symmetrical example. Let's look at a slightly less symmetrical example. And all we'll do is replace one of the chlorines with a fluorine. Okay, so that's the second example. We still have this sort of plane of symmetry here, where this side is the same as that side. But if you think about the other one we talked about, it's no longer the case. The 2 halves are no longer the same. So let's think about the different environments now. Well, let's start with this section here, these ones here. So we can say that these 2 are the same as each other because they're kind of near the chlorine. So that one are the same. Okay. However, these 2 are no longer in the same environment because these ones are nearer the fluorine than the chlorine. So we'll mark these a different colour. So they'll have a different environment associated with these. And then let's just consider the directly bonded atoms. Well, clearly, hopefully clearly anyway, that one is now different to that one, which is different to the previous example. So again, we'll use different colours. That's another chemical environment. And finally, what colour am I use? Green. That is also a different chemical environment. So we will expect to see a grand total of 4 signals for the less symmetrical example.
Okay, thank you, Nick. So what we're going to do now is go back to your widget and have a look to see what the answer is. So I'm going to put mine in here. So we can see that the majority are 69% are going for 6 in different carbon environments, and the remainder are going for 4.
Okay, so let's just have a look at what the answer is. So go through that. So we're just going to basically highlight on the sophisticated picture what we've got. So we have, that's probably one environment, the CH3's there. This is a second environment, the CH2 there. This will be a third environment. Those 2 together will be a 4th environment. Those 2 together will be a 5th environment. And lastly, there'll be a 6th environment. So those of you who got 6 were right, which is excellent.
Okay,
so the majority of you.
Shall we have a look? So what we're going to do now is we're going to have a look at the carbon-13 NMR spectrum.
Yep.
So shall I help you get rid of that?
Get rid of those things, the various pictures we've got. Okay, so let's pull across the carbon-13 NMR spectrum, okay, and run through this. And this is one of the ones that Matt very kindly produced for us for ethyl benzene. So first things first, I've marked in blue here the solvent. Remember, we use CdCl3. I don't want to confuse that with lines from the sample. So we get one, which is due to the carbon in deuterated chloroform. This one about 77, so that's that one there. So let's count the other ones. Well, we got one there, obviously up very high up. We've got two lines close together, which might be quite hard for you to see. There's actually two lines there. So that's another 2, 3. This is then the fourth, a little bit further along. And then we've got a fifth and a sixth. Okay, so we've got a grand total of 6 lines for that, which is what we'd expect. I should probably also mention what we'd expect to see on a before we're going to look at in a moment about sort of finding out what those lines correspond to. But when you have an NMR spectrum, along the horizontal axis, you've got the chemical shift, which is measured in parts per billion. Okay, million even, parts per million even, and it runs, and so the lower values are on the right-hand side, and the higher values are on the left-hand side. So for a carbon NMR, it runs from about 0 up to about 200 ppm. Okay, so that's kind of the general structure, and we'll look when we look at a proton NMR what they look like, which is slightly different but has similar features. So that's the first thing we want to do, and the next thing is to look at the correlation table.
I think so, but I think first of all we were going to make a quick note about the reference. So one thing that maybe wasn't obvious in the video of the actual experiment itself was the fact that there's a reference compound that will be included in when they do the NMR spectrum. Now the reference is set to have a chemical shift to 0, and everything else is basically measured relative to that. In this particular example, we're not seeing the reference, and in fact, I don't think we do in any of the spectra that we're going to see here. So the reference would be at 0, and some of these spectra are actually run or have been cut so they're slightly above 0, so that you don't have to get confused with having the reference peak within there. So in this case, it's no reference, which is why we've got the 6 peaks.
Yep.
Okay, right. So as you say, we're going to look now at the correlation chart. So in this case, this is the carbon-13 NMR correlation chart that you may be familiar with from level 2. This is provided on the module website under the assessment resources because you will need it for TEMA 03. Okay, so if we have a look at this, so obviously we were talking about the fact that we're going to have, we have aromatics, so we would expect to be having the aromatic carbons. If we look on the correlation chart, you can see that the aromatic carbons, hopefully you can see that, but hopefully you've got your own charts with you that might have a little bit more detail, but they occur between about 110-ish to about 150 ppm. So is that consistent?
So yeah, we've got 4 signals, as I think a lot of you saw, in the aromatic, and these are all above 120 and slightly below 150. So these 4 signals here would be the aromatic signals. Okay,
and then what we're seeing down here is likely to be due to the aliphatic. So that would be the CH2 here, the methylene, and the CH3 carbons. So these are, as I say, down here, the aliphatic or alkane are between about 0 and, I don't know, around about 55 ppm. So very much where we would lie here.
Yep.
Another key thing to note is that actually, even though we know that these are due to the aromatic, we can't really predict exactly which one is which, so which one applies to which carbon. I think that's fine when you're doing an interpretation. Quite often you can't assign something quite unambiguously. So down here we've got these two. So which one do we think is due to the methyl and which one is due to the methylene?
Okay, so I would go for the one around nearly 30 being the one that's adjacent to the benzene ring. And the benzene ring, as you may have seen, has a de-shielding effect. So these CH2s are being slightly de-shielded by their proximity to this benzene ring, so they pull to a slightly higher chemical shift. So this is likely to be the CH2, whereas the one a little bit further away, the one around 15, is likely to be the CH3.
Okay, and there's plenty about de-shielding. As I say, if you want to go and find out more about de-shielding and the influence of that on the chemical shift in the NMR, the fundamentals slide cast, so it talks about that there.